But we have already talked about some master regulators, such as a kinase called AMP kinase, which gets activated by a high amount of AMP. So a high AMP to ATP ratio will activate this kinase, AMP kinase. And what does it do? It will coordinately activate catabolic processes. What are those? It could be fatty acid synthesis. It will be inhibited. So instead, it will promote fatty acid breakdown. How does it promote fatty acid breakdown? One way of doing it is to activate mitochondrial biosynthesis. So the more mitochondria you have, the more you can break down fatty acids, for example. Also, AMPK will activate glycolysis. That's a fundamental catabolic pathway, central metabolism we have talked about. So it activates catabolic processes and at the same time inhibits anabolic processes. What are those? Fatty acid synthesis. We discussed this, how this happens. There's a phosphorylation on ACC1, which is the rate-limiting fatty acid biosynthetic enzyme, which inhibits it. So you don't make more fatty acids, you break down more fatty acids, for example, in mitochondria. So yeah. And so that's the general theme. That's how it is a general master regulator of catabolism. It activates catabolic process. That's one enzyme. And we have seen this slide before. AMP levels are high, AMPK will be high, and that triggers essentially processes that generate ATP and inhibit and processes that use ATP, such as biosynthetic processes, fatty acid synthesis, and so on and so forth. AMPK kinase will if you go by tissue and organ, it will activate things that work very hard, such as muscle. They rely on a lot of ATP. Muscle and heart, for example, are pumping all the time. So that's how those processes in these tissues will be activated. The brain also, it needs a lot of glucose, so glycolysis goes on in a massive extent. That will be activated too. And other biosynthetic organs will be, or storage organs and tissues will be inhibited. The process like fatty acid synthesis, for example, in adipose tissue. So now we see two fundamental types of metabolism, depending on whether the cell is proliferating and dividing, which means it needs biosynthesis, it needs anabolism, or if the cell is just sitting around and just being happy doing what it does, right? But without making new cells. In the latter case, the quiescent cell that doesn't divide, let's say a heart cell, the metabolism looks a lot like the type of things we discussed in the first half of the course, right? It does a lot of glycolysis, it does a lot of the TCA cycle, burns a lot of fatty acids. So these are the general pathways. But in a proliferating cell, and what are those? In normal individuals, this will be, in healthy individuals, this will be immune cells. So our immune system generates all the time new cells. Or of course, in pathology such as cancer, the proliferating cell, then what happens is, the metabolism looks very different. Look at the arrows. It's still the same pathways, but a lot of the glucose flux gets diverted to these magic intermediates, that will generate amino acids or other things through glycolysis. Likewise, the TCA cycle is still there, but some portions of it may run in different manner. For example, you import a lot of glutamine, you run this portion backwards, counterclockwise, up to citrate. Citrate gets out, you generate acetyl-CoA, and let's say you want to make more membranes, more lipid. So the pathways are the same, whether you're a quiescent or proliferating cell, but the flux that goes through this pathway is very, very different, right? And this is a more clear view of cancer metabolism, how it has been rewired. So a blow up of this lower part, the proliferating cell, you can see exactly what it likes to do. The little arrows that come off here you see pentose phosphate pathway to make ribose 5-phosphate. So you can make nucleotides, as we saw in the previous lecture. 3-phosphoglycerate, glycolytic intermediate. What will that do? It will give you serine, that we saw that in nucleotide, in amino acid biosynthesis. And of course, with the folates that we discussed, the serine will give one carbon to folate, will generate glycine, and the folate, the one carbon, the folate can be used to make nucleotides, as we discussed last time. For glutamine, as I said, glutaminolysis is very important in cancer cells. They pick up glutamine, and they run this portion of the cell of the TCA cycle in the reverse. You get to citrate comes out, and then you can generate acetyl-CoA, and from then on, from acetyl-CoA carboxylase, ACC, and fatty acid synthase, you can make lipids. And anything you see in blue, like FAS, IDH, these enzymes here, these are all now metabolic enzymes of central pathways that we have discussed in an undergraduate class like this, and they have been found to be mutated in various cancers, okay? Indicating that really the cancer cell, to maintain the proliferative status, has to rewire its metabolic pathways from this to something that looks like that, and that's why it activates these enzymes. So this has been the revolution, really, in biochemistry and metabolism as it relates to cancer. This realization of how important it is to not only sustain cell proliferation, but also drive oncogenesis in many cases. So, all right, so far so good about the master regulator of catabolism, which we said, of course, it's AMPK. But that's when you want to break down. So if you are working in this scheme, this is what AMPK activation will do. What about the proliferating cells? When you actually have biosynthetic needs and anabolic needs, is there a master regulator for this process, right? And that's obviously very important because, again, it's involved in all these pathologies such as cancer. So here we're going to take a little detour, and the answer is yes, there is a master regulator of growth, of biosynthesis. And we'll talk about the TOR gene, okay, TOR. And the story goes back to Easter Island, Rapa Nui. And, in the 70s, still today, actually, this day, but early on in those decades, major pharmaceutical companies will prospect. They will go to weird places all over the earth and scoop up some dirt and find useful microbes that maybe make some weird stuff with useful pharmaceutical properties. So Wyeth Labs at the time found something which looks, it's a lipid, it's a complex lipid made by microorganisms, by bacteria, and some streptomyces, I believe. So I've shown you the structure actually when we're talking about lipids. And that was originally identified as an antifungal. So the bacteria make it, presumably to kill fungi that attack them, in the soil of this island, the Pacific Island. The compound was called rapamycin from the island Rapa Nui. So originally it was found to be antifungal. Then in the 80s and 90s, it was used as an immunosuppressant. It had the immunosuppressant properties, meaning it inhibits proliferation of immune cells. And why would that be useful? As I've told you last time, in transplantation, for example, you want to suppress, in organ transplantation, you want to suppress the rejection of the organ by your immune system. So that was the original use of the drug, very popular. Then it was found it has anti-cancer properties. So it will actually stop cell division. And here we get to stop, you see this fundamental property of stopping cell division, whether it's a fungus, whether it's an immune cell, or whether it's a cancer cell, you add rapamycin and it will stop cell division or delay cell division. Then it was also discovered that it has anti-aging properties. And then you start wondering what else could it have? What other uses? But the race was on to find the target of rapamycin. How does it work? And for this, yeast, the lowly system, it is a eukaryote, but it's a single cell organism, is extremely helpful, as in many other cases. And the way to go about it is to find a mutant yeast that is immune to rapamycin, doesn't see the drug, it's not inhibited. Then you go and sequence the genome of that, or find in those days by more laborious techniques, the genetic association with that resistance to the drug, and try to find that gene. So the gene was identified in Mike Hall's lab, And it was shown that really what rapamycin does is to inhibit a protein kinase complex. So you have to imagine that there's a protein kinase that regulates cell division or cell proliferation in general and growth. So all these biosynthetic processes, and rapamycin goes and inhibits it, and it does that in all these organs, whether it's a fungus, a yeast, or whether it's a T cell, an immunosuppressant situation, or a cancer cell. So in all these cases, it blocks cell division by inhibiting this kinase. And many, many years went by. This was like 20 years later after the discovery of TOR. The gene TOR was found to, not only that yeast has the TOR gene, but actually yeast has two genes, as it turns out, but we have only one. And the kinase is part of a big complex, the TOR kinase complex. The X-ray high-resolution structure is still not available. It's a huge protein complex. But we have some electron microscopy data. It looks like this. And sure enough, again, after many, many, many years and enormous amount of work, you can see if you add rapamycin, rapamycin promotes the binding of an inhibitory protein shown here in pink. So you compare the unbound without rapamycin plus rapamycin, you see this little globe there. That this is what happens when TOR is inhibited by rapamycin. This is kind of beautiful, to go all the way from the genetics to identify the gene genetically, and then go to the structural information and how you see the structural difference, whether something is inhibited or not. It's quite beautiful. Now, needless to say, this is a major target for cancer therapy. There are many rapalogs, as are called, analogs of rapamycin that perhaps have improved properties. These days, anyone who has a heart attack and survives it and gets a stent, usually the stent will be impregnated with one of these rapamycin-like drugs. And you ask, why is that the case? Because the major reason of failing, a stent implant is because endothelial cells are proliferating around the stent and block it. So one way to block, to inhibit this endothelial cell proliferation in the vasculature is to impregnate the stent with rapamycin, so you suppress the cell division again. Same idea. So what does TOR do? I mean, TOR, as I said, it's a kinase. It's inhibited by rapamycin, and it does pretty much the opposite of what AMP kinase does. Okay, it promotes anabolic biosynthetic processes. How does it do that? It promotes protein synthesis by removing some inhibitors of protein synthesis. Protein synthesis, by definition, is something that a proliferating cell has to do a lot to generate new proteins for the newborn cell that it will give rise to. It will promote lipid synthesis. But how does it do that? By inhibiting an inhibitor of lipid biosynthetic genes. It will also promotes adipogenesis. It promotes all kinds of things. All right. So the general view is that you have two master regulators. One, AMPK, which is a master regulator of catabolism, and the second one, TOR, which is a master regulator of anabolisms. Here, the complex called TORC for TOR complex 1. Okay. And if you like a schematic, a different schematic, and we're going to talk about this again. Remember that TOR promotes anabolic processes, and AMPK promotes catabolic responses, catabolic processes. So is any of that related to aging? Well, why would we care? Now, the highest risk factors of most cancers is age, right? By the time, now you don't care because you're somewhere, let's say here in this group, so your cancer risk is very low. I'm right here, so I'm looking at the abyss, and as you know, a family member is facing this calamity. So, and this applies to many other diseases, not just cancer. Okay, you take diabetes, you take whatever you want to take, and all the other risk factors are tiny if you compare them to the risk factor of age. So the longer you stick around, the higher the probability you're going to get any one of these diseases, including cancer. All right, is there anything you can do about it though? And until very recently, and this I took this slide from 2005 from the World Health Organization, the answer was no. And by the way, if you talk about chronic diseases like, as I said, diabetes, cancer, and all these other things. These are the major killers you can of not just developed societies, industrial societies, but on a planetary scale, right? So if you ask how many people die from, famine, war, infectious diseases, or whatever, you put them all together, nothing comes even close to the death rate from chronic diseases. So clearly, chronic diseases, and they seem to be all be associated with a higher risk in the older you get, all right? All of them are the major killers. And there are some things you can do. So this is a risk factor. Obviously, age is a risk factor, but is it a modifiable risk factor? I mean, smoking is a risk factor for lung cancer, but there is something you can do. You just quit smoking, right? Is there anything you can do about more advanced states? And that was considered to be an unmodifiable risk. So there you go, AIDS, okay? Likewise, for your genes, your genetics, you can't change them. So that's not really easily modifiable unless we have some gene therapy which raises some other issues. So this was as of 2005. But is really aging a modifiable risk or not? It was thought it is not, but one of the triumphs of modern aging research is to convincingly show that it is very modifiable, actually. So go back to yeast now. Again, the very first evidence that aging is a modifiable risk factor also came from yeast and other lower systems such as worms, for example. How can you measure aging in yeast? Well, you can, the cell is born, it will make a little bud, which will give rise to a daughter cell. That's a normal cell division, and then you end up with a mother cell. Well, it turns out that the mother cell will keep on dividing. Not forever. No cell is immortal. It has a finite number of times it can divide, usually about 25 in yeast. And by the way, even human cells, you take them out of the tissue, you put them in a culture dish, they will divide about 30 times or so. So no cell is immortal, and the same goes for yeast. The population of yeast cells is immortal because you keep generating the young ones. So it was shown, back to rapamycin now, in 2005, that rapamycin and TOR regulation, okay, promotes aging. So either you add rapamycin or you inhibit it, you delete, you mutate, let's say, the TOR gene in yeast, that will make yeast live longer. And the same results were found in many other organs, okay, after that, in worms, in flies, and so on and so forth. So that seemed interesting. So remember now, we have a master regulator of growth that promotes cell proliferation, but some moderate, at least, inhibition of it makes the organism live longer. And now the race was on to identify the magic drugs that will make you live longer, not forever, but longer, and importantly, to live healthier. Okay? So an objective of aging research is not necessarily to make you live longer, it's to increase your health span so you have more years disease-free, essentially, before you die. So to push all the onset of all these chronic diseases as late as possible. So this is the evidence. What you're looking at here is rapamycin, the magic drug we have been talking about, when given to mice. This is now going from yeast to mice. That's a huge jump, right? Mouse is a mammal. Many pathologies very similar to human diseases, and you add, here is an even more remarkable finding. Not only rapamycin, here you see a survival curve, so age in days on the x-axis and percent survival on the y-axis. The moment you give rapamycin, these mice were already very old. I mean, they were middle-aged, okay, so 600 days old, so a little over a year old. They live about 2 years, 2 to 3 years at most. And this is, the blue is without, the red is with rapamycin, and you give the drug, to a 60-some-year-old equivalent to human years mouse, and now this mouse can live longer and healthier. So that was an amazing experiment, right? A huge experiment. I should say that the same results now have been are ongoing big studies in dogs. So why not humans, right? I mean, so until last year, there was no the major hurdle was that the FDA would not approve a drug to improve longevity or delay aging, because FDA only approves drugs for a given indication, for some disease. Okay, aging was not considered a disease, even though it's a risk factor for all these other chronic diseases. Aging itself is not considered a disease, so there was no way to get even clinical trials going. Now there is one that is happening in humans with a different drug that's also quite promising. But another avenue that the researchers have taken, some of the same researchers that did all these experiments, actually, you see some of them here. The first author and the last author was to, Matt Cabernet and Brian Kennedy, is to start clinical trials in dogs, in companion dogs, not experimental dogs, like people's pets, okay? Because they all come in many different genetic backgrounds, just like people. They live in the same environment as people. Okay, I mean they interact with people all the time. So as long as you have the consent of the owner, these dogs are taking a daily pill of rapamycin, and you should know that ANM is a big part of that study, especially for the effects on their circulation, the heart function, and the results are extremely good. They actually, rapamycin in dogs improves their health span. I mean, their cardiac function is much better compared to without rapamycin, and it's still too early to know if it will extend the lifespan. But it certainly does help with some health span markers such as heart function. And I show you some evidence we had, I don't know, we're talking about tryptophan and tryptophan biosynthesis and ibuprofen. I had told you how ibuprofen extended the lifespan of yeast and the worms in our lab. And ibuprofen, turns out, is, it's a super simple drug. It's on the World Health Organization list of core drugs. And we had found this, okay, that give ibuprofen to yeast and worms and other systems, and they will live longer, just like the rapamycin. How does it do it? It turns out that the very first substrate of rapamycin that was shown to, or downstream effect of rapamycin that was identified ever in the literature in yeast, was to promote the destruction of this tryptophan transporter. Remember, tryptophan is very expensive to synthesize, so yeast, even though it can synthesize it, it prefers to take it from the outside. So what we found is that what ibuprofen really does is to promote the degradation of this transporter that brings tryptophan in, so the cell effectively has less tryptophan available to it. So it's this tryptophan limitation that promotes lifespan. And rapamycin, some of its effects actually come from that also, because rapamycin TOR normally blocks the degradation, but ibuprofen does the opposite, as I said. So if you add rapamycin, you inhibit the inhibitor of tryptophan transport, so you activate, so you really fail also to bring in tryptophan.